Crossing over typically occurs between chromosomes as gametes are formed. In this animation, you see this crossing over occurring normally, and each chromosome has its same gene content after the crossing over is done. However, in the second animation, you see a second possibility. Sometimes the crossing over occurs unequally, so that the two chromosomes end up with differing number of genes. Perhaps they don't line up well at the start. So here, one chromosome has two gene copies where originally there was only one. If there were two Hox genes in a row, this is the situation which is true of cnidarians, then most of the crossing over events for gamete formation would have produced two genes on each chromosome. Okay. However, once again, there could be cases where abnormal crossing over resulted in one of the daughter cells getting only one Hox gene, and a second one now getting three in a row. And thus unequal crossing over could cause this small Hox cluster of two genes to amplify to three. A Hox cluster with three genes would undergo crossing over during gamete formation, and most of the time this would leave each cell with the normal set of three Hox genes in a row. However, once again, abnormal crossing over events could happen, so that one daughter cell would receive perhaps four Hox genes in a row, while one received less. So thus unequal crossing over could cause this Hox cluster, this series of Hox genes in a row, to amplify further as more genes are added as copies of original genes. Obviously, this process could continue. Worms may have three or slightly more Hox genes in a cluster, but by the time you get to the higher animals, they have far more. It seems that these rows of Hox clusters were gotten by unequal crossing over, duplicating Hox genes from smaller ancestral structures, so that there would be an anterior group here in pink, a medial group here in blue, and a posterior group here in aqua. So from a small number of genes, ultimately one, unequal crossing over would produce a cluster.